At ASCO 22, there were presented results of three very interesting trials. Tisotumab vedotin, a new antibody drug conjugated in combination with pembro or carboplatin in advanced cervical cancer. Trabectedin versus physician choice chemotherapy in recurrent BRCA-mutated or BRCA-ness phenotype ovarian cancer. And lastly, ATHENA-MONO with the results of rucaparib, a single agent in the maintenance setting of high-grade serous and endometrioid newly diagnosed ovarian cancer. During ASCO 2022, I presented the results of TB 0.5 trial, which was a phase 1b trial with an expansion cohort addressing the value of tisotumab vedotin, a new antibody drug conjugated directed toward tissue factor, in combination with carboplatin in untreated metastatic patients, or in combination with pembrolizumab in untreated metastatic patient, or in combination with pembrolizumab in previously pretreated patient. This drug seems to be very interesting. It has been already approved as a single agent in the United States based on the result of a single-arm phase 2 trial reporting 24% response rate. What we did in our trial is to evaluate the possibility to combine with carbo or with pembro. In the cohort of untreated patients in combination with carbo, a very interesting signals of activity with 55% overall response rate was reported. In the unpretreated cohort in combination with pembro, 40% response rate was reported. In the previously pretreated cohort in combination with pembro, 32% overall response rate was reported. Very interesting, the toxicity profile of the drug was quite manageable. As for every ADC, the particular toxicity is related to ocular toxicity, peripheral neuropathy, and also neutropenia, but there were mainly grade 1, 2, and grade 3 toxicity under 5%. So, a new drug with a huge possibility in the algorithm of cervical cancer and the possibility to combine with platinum and pembrolizumab and to move forward in the first line. At ASCO 2022, there were presented results of a randomized phase 3 trial called MITO 23. It is an academic trial which randomized 244 patients to receive trabectedin single agent versus standard of care, which was represented by 5 different options at physician choice between carboplatin, weekly paclitaxel, pegylated liposomal doxorubicin, topotecan, and gemcitabine. Trabectedin has been already approved in Europe in combination with pegylated liposomal doxorubicin for the treatment of platinum-sensitive recurrent ovarian cancer patients, but preclinical and early clinical data suggest a peculiar efficacy of trabectedin when used as a single agent in BRCA-mutated or BRCA-ness phenotype patient. And this is what we want to address in this trial, if trabectedin single agent is superior to standard of care. The primary endpoint was overall survival, but unfortunately the trial did not meet its primary endpoint, and trabectedin did not report to increase overall survival with respect to standard of care, and also secondary endpoints were negative. And there was no significant benefit in terms neither of progression-free survival or overall response rate. The toxicity profile of the drug was confirmed. Trabectedin was more toxic than standard of care in terms of neutropenia and neutropenic fever, fatigue, hepatic toxicity, and gastrointestinal toxicity. So, the drug reported retained clinical activity in this so heavily pretreated population. More than 70% of patients have received at least 3 prior chemotherapy lines. 35% of them have already received PARP inhibitors. So, the drug retained activity, but it was not superior to standard of care. At ASCO 2022, we present the results of ATHENA-MONO, which is a large randomized phase 3 international trial comparing maintenance treatment with the rucaparib versus placebo in a population of stage 3-4 high-grade serous and endometrioid ovarian cancer. To put this trial in the context, we already realized that maintenance treatment with PARP inhibitor increased progression-free survival, firstly in BRCA-mute patients. Then we broadened the indication also to non-BRCA population, and actually we have 2 PARP inhibitors already approved in the first-line setting. And now we have the results of a third PARP inhibitor because rucaparib met its primary endpoint, and a significant increase in progression-free survival versus placebo was reported in patients who received rucaparib maintenance treatment for 2 years after completion of chemotherapy. The magnitude of benefit was higher in BRCA-mute patients, was intermediate in BRCA wild-type HRD positive population, but also HRP proficient population experienced a significant increase in the median progression-free survival when receiving rucaparib as maintenance treatment after completion of chemotherapy. So the results of this trial confirm the benefit of maintenance treatment in first-line setting with PARP inhibitor. The toxicity profile of rucaparib was really superimposable to what we have seen in second line. Mainly the toxicity was related to hematological toxicity and anemia in particular, but there is a key toxicity of rucaparib which is represented by increases in transaminases. We cannot call them hepatic toxicity because bilirubin was not increased, so we will call them increase in transaminases, which was 11% in 11% of patients, but very manageable by reducing the dose. In fact, in the trial, around 50% of patients reduce at least one level of dose in order to manage toxicity.